Environmental DNA, eDNA, and Mendel. What is environmental DNA? Environmental DNA identification has become critical in performing environmental science research relating to both endangered species and invasive species. Environmental DNA is nuclear or mitochondrial DNA that is released from an organism into the environment. Sources of eDNA include feces, mucus, gametes, shedding skin, hair, and carcasses. In aquatic environments, eDNA is diluted and distributed in the water, where it persists for a maximum of 7 to 21 days. What is DNA? Deoxyribonucleic acid is a molecule that carries most of the genetic instructions used in the development, functioning, and reproduction of all known living organisms and many viruses. To understand the history of DNA's role in scientific research, we must travel back to the early 1800s in Austria. The work of Gregor Mendel. Every little thing has characteristics inherited from its parents. Genetics is the scientific study of heredity, or the process of inheritance. Gregor Mendel's peas. An Austrian monk named Gregor Mendel studied science and mathematics and taught at his monastery's high school. Mendel was in charge of the monastery garden and performed studies with pea plants. Fertilization of pea plants. The male portion of the flower produces pollen, which are the male reproductive cells. The female portion of the flower produces egg cells. Fertilization is the joining of the male and the female cells. True breeding. The stalks of pea plants were true breeding, which means that they would always produce the same types of plants if they were allowed to self-pollinate. An allele is a specific form of a gene. True breeding means all plants have the same allele, which is the alternate form of a gene. So the stocks that Mendel had were true breeding, which meant they had one type of allele for each characteristic. True breeding. For example, all the true breeding plants which self-pollinate would produce green seeds, or all would be tall. Self-pollination. Normally, pea plants will self-pollinate. Cross-pollination. Mendel wanted to produce seeds by joining male and female reproductive cells from two different plants. To control this, Mendel cut off the pollen-bearing male part of the flower. He would then dust the female part of the flower with the desired pollen from another flower. This process allowed him to control the crossbreeding of plants which had different characteristics. Genes and dominance. Mendel studied 7 different pea plant traits. A trait is a specific characteristic, such as seed color or plant height, that varies from one individual to another. Each of the 7 traits that Mendel studied had 2 contrasting characters. For example, green seed color and yellow color. Mendel crossed plants with each of the 7 contrasting characters and then studied the offspring. The original plants are called the pea or the parentals. The offspring are the filial. The first offspring was F1 or first filial. The offspring of crosses between parents with different traits are called hybrids because they have both of the 2 forms of the allele. Genes. From his experiments, Mendel concluded that inheritance is determined by factors that are passed from one generation to the next. We call these factors genes. Dominance. Mendel also concluded that some alleles are dominant and some are recessive. An organism with the dominant allele for a trait will always exhibit that trait, or the dominant trait. Recessive. An organism with the recessive allele for a trait will only exhibit the recessive trait when the dominant allele is not present. Segregation. The F1 cross. Mendel allowed the F1 plants to self-pollinate, in effect, crossing the F1 with itself in order to produce the F2 offspring. 1/4 of the offspring exhibited the recessive allele when he viewed the F2 or filial 2. Explaining the F1 cross. Each F1 plant produces the two types of gametes. For example, those with the allele for tallness and those with the allele for shortness. Mendel explained the return of the recessive traits in the F2 by stating that the allele for the contrasting traits separated or segregated during the formation of gametes or sex cells. Each gamete carries only a single copy of each gene. The alleles separate during gamete formation, but then pair up again in the F2 generation. That is why recessive traits disappear in the F1, only to reappear in 1/4 of the F2 generation. Probability and Punnett squares. Whenever Mendel performed a cross, he always counted the offspring of the parental P-cross, which were the F1 and the F2 generations. When he crossed two F1 plants, which were hybrid for tallness, heterozygous tall crossed with heterozygous tall, 1/4 of the F2 product would be recessive T trait, and 3/4 of the F2 product would be the dominant T trait. He realized that probability could be used to explain the results of genetic crosses. Genetics and probability. The likelihood that a specific event will occur is called its probability. The probability of a coin flip resulting in heads equals the number of actual outcomes over the number of possible outcomes, or the number of ways it can happen. In other words, heads, there's only one way that heads can happen, and the total number of possible outcomes. The total number would be heads and tails, so that would be 2. So that is 1/2. To calculate the probability of flipping 3 heads in a row, because they are separate events, simply multiply the probabilities. 1/2 times 1/2 times 1/2 equals 1/8. So the probability of flipping 3 heads in a row would be 1/8. The fact that you multiply the probabilities together illustrates an important point. Past outcomes do not affect future outcomes. Punnett squares. The gene combinations that result from a genetic cross are determined by drawing a table called the Punnett square. This Punnett square shows one of Mendel's experiments where two plants which are hybrid or heterozygous for tallness are crossed. Heterozygous tall crossed with heterozygous tall. The gametes produced by each of the two F1 parents are shown in the top here and on the side of the diagram. So heterozygous tall here is crossed with heterozygous tall. So these are the two parents. And these are the resulting offspring. Punnett squares. The possible gene combinations of the F2 offspring are shown inside the boxes. The letters within the Punnett square represent alleles. T represents the dominant allele for tallness, and lowercase t represents the recessive allele for shortness. Punnett squares can be used to predict and compare the genetic variations that result from a cross. So notice we have the uppercase T representing the dominant tallness, and the lowercase t representing the recessive allele for shortness. Notice that we use the letter of the dominant trait to represent the characteristic. So in this case, tallness is the dominant trait. Therefore, we use T to represent tallness and lowercase t to represent shortness. Homozygous versus heterozygous or hybrid. Homozygous, organisms that have two identical alleles for a particular trait. That would be homozygous dominant, tall, or homozygous recessive, tall. Heterozygous, organisms that have two different alleles for the same trait. Homozygous organisms are true breeding for a particular trait. Heterozygous organisms are hybrid for a particular trait. So here we have homozygous dominant, here we have homozygous recessive, and here we have heterozygous tall. So this is actually the hybrid. All of the tall plants have the same phenotype or physical characteristics, but they do not all have the same genotype or genetic makeup. The genotype of 1/3 of the plants are homozygous, homozygous dominant, or the genotype of 2/3 of the plants are heterozygous or hybrid. Probability and segregation. If you look at the F2 offspring, 1/4 of the F2 plants have two alleles for tallness. This is homozygous dominant. 2/4 or 1/2 have one allele for tallness and one allele for shortness. So 1/2 are actually hybrids. Because the alleles for tallness is dominant over the allele for shortness, 3/4 of the F2 plants will be tall. So their phenotype is tall. Overall, there are 3 tall plants for every 1 short plant, or a ratio of tall to short 3:1. Because experiments fit this model, the predicted ratio of 3:1 tall to short occurs, thus supporting Mendel's belief in segregation of alleles. Here we have the parental generation. Notice this is homozygous dominant, this is homozygous recessive. Those are the assumed parents. So the offspring from the homozygous parents will be heterozygous, and they will all be hybrids. They will all be heterozygous, tall. So that's the F1 generation, or the filial one. And then when we pollinate or self-pollinate the F1, we end up with a different ratio. Notice we have a ratio, we end up with 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So this is the F2 generation. Probabilities predict averages. Probability can predict the outcome of a large number of events, but probability cannot predict the specific outcome of one individual event. The larger the number of samples, the closer we get to the predicted outcome. Exploring Mendelian genetics. After showing that alleles segregate during the formation of gametes, Mendel wondered if they did so independently. In other words, does the segregation of one pair of alleles affect the segregation of another pair of alleles? Does the gene that determines whether the seed is round or wrinkled have anything to do with the gene for seed color? In other words, are the genes somehow linked, or are they completely independent and segregated? Independent assortment. To answer this question, Mendel performed an experiment to follow 2 different genes as they passed from one generation to the next. Mendel's experiment is known as a 2-factor cross. So we're looking at 2 different genes. The 2-factor cross, the filial one. First, Mendel crossed true breeding plants that produced only round yellow peas, the genotype homozygous round, homozygous yellow, with plants that produced wrinkled green peas, homozygous wrinkled with homozygous green. All of the F1 offspring produced round yellow peas. This shows that the alleles for yellow and round are dominant over the alleles for green and wrinkled. So therefore, Yellow and round are dominant. Green and wrinkled are recessive traits. A Punnett square for this cross shows the genotype for the F1 plants are all heterozygous for color and heterozygous for roundness. The two-factor cross, F1. This cross does not prove that the genes assort. It merely provides the hybrid plants for the next cross. So notice we begin with homozygous dominant crossed with homozygous recessive. The gametes, these are the gametes that are produced. And the F1 are all identical. They're all heterozygous for color and heterozygous for roundness. The two-factor cross. Mendel knew that the F1 plants were all heterozygous for seed shape and seed color. But how would the alleles segregate when the F1 plants were crossed with another to produce the F2? So we're going to take this, the heterozygous, and we are going to basically self-pollinate and then find out what we end up with the F2 generation. Mendel's experiment, the F2 generation produced 556 seeds, and they were in the ratio of 9:3:3:1, demonstrated by the Punnett square. So here we see we're actually counting the number of dominant. So we have 9 that have the dominant trait. We have 3 that have the 3 that have the heterozygous, 3 that have the heterozygous, and then 1 that is homozygous recessive. So we end up with the ratio in the F2 of 9:3:3:1. So that is a phenotypic ratio. Independent assortment. This proved that the alleles for seed shape segregated independently of the alleles for seed color, a principle known as independent assortment. The principle of independent assortment states that genes for different traits can segregate independently during the formation of gametes. Independent assortment helps account for the many genetic variations observed in plants, animals, and other organisms. So this actually proves that the genes are assorting independently. Summary of Mendel's principles. Mendel's principles are the basis for the modern science of genetics. The principles are: the inheritance of characteristics is determined by individual units or genes. Genes are passed from the parental to the offspring. Second, when there are 2 or more forms or alleles of the gene for a single trait, some may be dominant and some may be recessive. Third, in most sexually reproducing organisms, each adult has 2 copies of each gene, 1 from each parent. These genes segregate during the formation of gametes. And the final point, the alleles for different genes usually segregate independently of one another.